So we talked about the food consumption data, and we move now to the second pillar of let's say, exposure assessment, which is the food occurrence data. When we talk about food occurrence data, we are talking mainly about what the food contains as contaminants, and this is mainly to measure the concentration of the hazard of the mycotoxins, heavy metals, pesticide residues, etc. How to build it? It's really to focus on staple food and high-risk commodities, for example, aflatoxin and nuts. To have national monitoring plans that can have systematic sampling and analysis across the supply chain, and to invest in accredited labs with validated methods in order to improve the capacity of the conformity assessment and listing. Obviously, having centralized data repository is very important as a national database to store, manage, and share, obviously, after analyzing the results to all, let's say, communities concerned with the risk assessment. There is a lot of food contaminants database that we can mention, mainly the GEMS to estimate the chemical concentration. And here we have a screenshot of this database where we can find for each contaminant a lot of information related to the occurrence date. We should know that the determination of the hazard of the food combination, it's very important for the risk assessment. We need to know exactly which agent is capable of causing health effects, and can this agent be present in food or not. Obviously, the data for hazard assessment can be experimental with the studies performed according to the international and the national guidelines. And this data about dietary intake for the dietary exposure assessment should be always reflecting the national occurrence and also the international dietary intakes at work. And this is the example of the assessment for the chemical. In brief, we should say that in the Arab region, there is always an objective to enhance codex capacity, and this is basically through the availability of data about, obviously, occurrence. There is always this discussion about the challenge of availability of accurate information, and this is why there was recommendations in order to make the data available for the community, the scientific community, through technical tools. This is why I'm presenting now the initiative on the Arab Food Occurrence Database, where we are collecting occurrence data through systematic scanning and data mining of published articles in order to synchronize it with the National Food Occurrence Database, and providing the data from different sources in order to be able also to contribute to the GEMS food database with an online portal that is mainly based on data mining for published and unpublished data to generate, let's say, some information about the control laboratories results, research institutions, and other, let's say, sources of information. So without further details, this is the proposed methodology. By data mining of published data, we can retrieve the information about the occurrence data, and we can work on it in order to have the reference, the contaminants, the food commodity tested, the study scheme, the analytical method, obviously, and the contaminant occurrence. And this will help us to have a certain level of benchmarking between the studies and the available information in order to see if they are, let's say, in a parallel level of benchmarking, in order to
benchmarking between the studies and the available information in order to see if they are in a parallel in order to be able to make it comparable. So this is some screenshots of database where you can find a lot of information about the occurrence data, with the LEQ and so on. And this is based on an online portal that can be accessed from anywhere from the world. You can have a search based web online portal by country, by food category, by food category, and even by year to know what's happening. So this is powered and under license from IT platform. It's mainly a platform, secure platform to log in and sign in in order to get access, or to the data entry part for the data, let's say visualization, where you can have some information for users, level of occurrence by country, by topic. So yes, we need to build an institutional framework, but what is needed practically? We are really searching to have a dedicated risk assessment every country, an independent scientific committee free from political and commercial influence, with a clear mandate and a legal framework that empowers the body to request data and issue scientific opinion. We need an interagency collaboration between the food control authority to provide monitoring and surveillance data, public health ministry, providing epidemiological data on foodborne illnesses, and agricultural ministry for pre-harvest information, as well as the national office to support the consumption survey designs. And this can lead obviously to create a sustainable ecosystem for risk assessment. Today we are calling for coordinated action to adopt common methodologies, to have standardized protocols for sampling, for analysis, dietary surveys, to ensure the data comparability. And this is a main challenge. Also, we are calling to establish a regional data sharing such the initiative we mentioned, a secure portal for aggregation, the occurrence and the consumption data, creating a regional risk assessment tool. And those will be supported by panels of risk assessment, joint scientific panels under existing regional bodies to address several issues and to sustain the capacity building. Obviously, a center of excellence or a regional training network to ensure the pipeline of skilled risk assessors is a must. Finally, we need to integrate with early warning systems to link risk assessment data with regional food safety emergency response faster and more targeted recalls. As a conclusion, the journey toward risk assessment in the Arab region is well underway. And the foundation is the structure, institutional framework, and the blueprint is provided by pioneering national experience and partnerships. By working towards harmonized approaches, we can collectively enhance facilitate trade and build a more resilient, safe food system for all. As a take-home message from what I presented today, yes, food safety is not about public health, but also economic stability, not only in the region, but all around. And risk assessment is grounded in scientific evidence is essential for effective food safety governance. The critical foundation is dietary exposure assessment that needs to be based on data on what people are eating and what contaminant hazards are present in food. Initiatives like the Arab food consumption data and the Arab food occurrence data are regional efforts to standardize and collect such data. The institutional capacity is key to translate data into actionable risk assessment, and regional efforts emphasize embedding exposure assessment within national food safety authorities to support evidence-based regulations. Yes, science-based today will ensure safe food tomorrow. Thank you.
Dr. Yazbek for this insightful and comprehensive presentation. Our second speaker is Ali Khalloud from Pereira, Laval University, Canada. Mr. Khalloud will present on the material and provide insights from the finding of exposure assessment.
From the Arab League countries, food safety decision-making and codex engagements. Mr. Balking and codex engagements. Mr. Balout, the floor is yours.
Assalamu alaikum wa rahmatullah. So thank you, Dr. Ruba, for your introduction. It's a pleasure to present the result of the first part of my PhD research at Université Laval under the direction of Professor Godefroy and co-supervision of Professor André El-Khouri. My name is Ali Balout, and today it's a pleasure to present the result of the first part of PhD research at Université Laval. Depending on several factors such as temperature, humidity, and physical damage of the crops. One of the most studied group of mycotoxins are aflatoxins due to their harmful effects on human health. So according to JECFA, aflatoxins are considered among the most potent mutagenic and carcinogenic substances known. They mainly contaminate 6 categories of food: cereals, nuts, spice and herb, and dried fruits and vegetables, coffee, and dairy products. Among all types of aflatoxin, AAV1 is recognized as the most toxic and prevalent form. The toxicity of aflatoxin B1 can be translated into 2 types of effects, depending on the dose and frequency of exposure. So in case of acute exposure, aflatoxin B1 may cause vomiting, diarrhea, abdominal pain, and even cerebral edema. In terms of chronic effects, aflatoxin B1 can lead to serious long-term effects, especially liver cancer, as well as immunosuppression and low birth weight. To limit this effect, we need to apply a combination of operational and regulatory approaches to reduce contamination. The operational measures include varietal selection, good agricultural, drying, and storage practices. On the other hand, we have the regulatory approaches. They are based on a risk analysis, which include 3 main components: risk assessment, which is the focus of this work, risk management, and risk communication. Now, the countries covered in this study are the 22 member states of the Arab League, located mainly in the MENA region, with a population of around 500 million people. These countries are particularly affected by mycotoxin contamination, mainly because of their climatic conditions. In order to assess the situation in this region and to develop an exposure profile, a three-step methodology was applied. First, we used a data mining approach to collect published data of aflatoxin B1 in the three main cereals, rice, maize, and wheat. Then a meta-analysis was performed to combine and harmonize the data obtained. Third, we conduct the risk assessment. In this step, we calculated the EDI, the estimated daily intake, to estimate the daily amount of aflatoxin ingested through the consumption of the three cereals. Then we performed a risk characterization by calculating risk of HCC, hepatocellular carcinoma, which provide an estimation of the number of liver cancer that could be caused by exposure to AFB1. We also calculated the MOE, the margin of exposure, which gives an indication of the severity of the situation. The lower the MOE, the higher the concern. Regarding the results of data mining, a total of 414 articles were screened and 35 studies met the inclusion criteria for the meta-analysis. Among them, 23 studies include samples of wheat, 13 of maize, and 10 of rice, with a total of nearly 3,000 samples analyzed. As mentioned earlier, these results were combined using a meta-analysis approach to obtain national and regional mean concentration for all three types of cereal. Moving to the risk assessment results, after calculating EDI, MOE, and risk HCC, we observed that the MOE values under both lower and upper bound scenarios were well below the EFSA safety threshold of 10,000. Also, the risk, the HCC risk, were higher than the limit established by WHO. This indicates the situation in the region could be concerning. At the national scale, the same pattern was observed, with the lowest MOE values found in Egypt and Iraq, where the situation appeared to be the most serious. Our findings should be interpreted with caution because the cereals are not the only source of AFB1. Other food products also contribute to exposure. We should keep in mind that these results are based on published studies, and studies reporting no detection are less likely to be published in the field. So the real situation could be less concerning than we currently observe. Also, this issue is not limited to the Arab region. EFSA has conducted a similar risk assessment for Europe, where the situation can also be considered as worrying. To conclude, the study combined data from 35 publications covering 8 out of the 22 Arab League countries and confirmed that the situation at the regional scale is concerning. These results provide a scientific basis to support risk management decision aimed to protecting public health and the regional economy. The main limitation of this study is the lack of available data. In terms of food consumption, official national data were often unavailable, and in terms in contamination, there was
Thank you, Mr. Ballout, for this excellent and very informative presentation. Our final speaker is Dr. Mustafa Sultan, representing GFORCE in the Near East region. Dr. Sultan will discuss harmonizing import maximum residue limits for pesticides, highlighting regional challenges, the need for coordinated approaches and alignment with codex and international standards. Mr. Mustafa, the floor is yours.
Thank you, Dr. Ruba, for the introduction. Good afternoon, esteemed colleagues. It's a privilege to join you at the 4th Global Food Regulatory Science Symposium here in Dubai. This presentation explores a scientific and regulatory pathway towards harmonizing of import maximum residue limits, MRLs, for pesticide residues across the Arab region. The aim is clear: to protect consumers while facilitating fair and efficient trade. Today's talk, we will move through 3 key parts: the background and rationale for harmonization, a detailed case study, and a proposed framework and call for regional collaboration. Let us begin with a quick background why pesticide residues are such an important topic for regulatory science and international trade. Before that, I would like to thank Dr. Eli for making this presentation much, much easier. Pesticide residues are at the heart of food regulatory decision-making. They are more than laboratory outputs. They influence market access, consumer trust, and the public health policy. Differences in MRLs between countries, or even the absence of an MRL, can become unintentional barrier to trade. Sometimes, a pesticide used safely in one country may not be registered in another. When that happens, shipments can be detained or rejected even if the food poses no health risk. To bring scientific consistency to these decisions, the Codex Alimentarius Commission has set a clear guidelines. Codex relies on the work of the joint FAO-WHO meeting on pesticide residue, which is the JMBR, which is an expert body that assesses residues based on toxicological and agriculture data. GMBR evaluates both acute and chronic exposure compared to a health-based guidance value, such as the ADI, which is the acceptable daily intake, the amount that can be safely consumed over a lifetime, and acute reference dose, which is the amount safe to consume within a single day. These guidelines also account for results related to patterns of use, fate of residues, analytical methods, etc. GMBR also reviews these values periodically, ensuring they reflect the most current science and analytical methods. But what happens when there is no Codex MRL and no national standard in place, or even when a national standard MRL exists, but enforcing it may compromise the food security consideration? This is exactly where the concept of an import MRL becomes essential. Now imagine this scenario. A food shipment arrives at the border, a pesticide residue is detected, but there is no local MRL, and Codex has not established one either. This creates a regulatory vacuum. The product is detained and the competent authority has no clear benchmark for action. In such cases, an import MRL or import tolerance provides a practical science-based solution. Before moving forward, it's important to define the term import MRL. As you can see on the screen, an import MRL is MRL set only for imported products, designed to meet trade and food security needs when neither national nor codex limits exist, but national food safety standards is achieved. This mechanism can also apply in cases where a food security concern arises even when an MRL exists, the product is of high economic value, or the pesticide is authorized abroad but not domestically registered. Import MRLs ensures that safety assessment remain consistent while allowing trade to continue without compromising consumer protection. To make such a decision consistently, a structured process or a decision tree is needed. The decision tree offers a transparent framework for evaluating import MRL requests. Let's walk through it step by step. As you can see, the first step is that the residue level non-compliant with national MRL or no codex MRL. So the first question we should ask is, is this level authorized in the exported country or by the exported national authority in this country? If no, then the answer is to reject the product. But if yes, we should request for the scientific justification for such registration or legal limits. So if justification is submitted, so we should review these scientific justification. Is it according to acceptable practices, transparency and risk analysis based? If no, then we reject the product. But if yes, then we should conduct a rapid risk assessment based on the national consumption patterns to confirm suitability, even if suitable or not. If the risk assessment conclusion shows a health risk, then we can reject the product. But if yes, then we accept the MRL as only import MRL, which is not designed for the domestic use, but only for the imported products. This approach would give us the opportunity, and this system would allow regulators to make defensible, evidence-based decisions while supporting trade continuity. With this framework in mind, the next step is to explore how a practical import MRL list can be established. A dedicated import MRL list would transform case-by-case decision-making into proactive, coordinated approach. Such list could be built by combining historical compliance data, systematic review of partners' practices, and industry-submitted justification supported by data from that decision tree. It's essential to clearly separate pesticide registration, which involves domestic use, from import MRL, which only applies to imported goods. This structured approach would allow competent authorities to review and update MRLs periodically using real trade and laboratory data. The result is a living system that ensures fairness, predictability, and transparency, attributes critical for both industry and regulators. Now, the foundation is clear. We can move to a real-world example, a case that illustrates exactly how this process works in practice. Our case study is about a practical case study about the scenario of tricyclazole residues in imported rice. And here we emphasize again on imported. We all know that rice is a significant contributor in our dietary habits in the Arab region, and any disturbance in the supply chain may consider as a food security issue. A shipment of rice was detained at the point of entry after laboratory analysis revealed that tricyclazole residues exceeded the national MRL. The detected levels was roughly 0.2 ppm. In this case, as we mentioned earlier, we can assume that no codex or national MRL or even the applicable MRL is 0.1 ppm, which means the consignment was technically non-compliant. However, all other pesticides tested in that shipment were well within the legal limits. Because of this single exceedance, the shipment was held under investigation pending a health risk assessment. Tricyclazole is a fungicide primarily used to control rice in rice blast disease, a major problem in paddy cultivation. It has been registered for agriculture use in several Asian and Latin American countries, including India and Brazil. However, it's not approved in the European Union, the United States, or Canada, primarily because of different regulatory philosophies and data requirements, not necessarily because of different toxicological evidence. As you can see, multiple jurisdictions have authorized the use of tricyclazole with an MRL 3.0 ppm, such as Japan, Brazil, New Zealand, and Australia. The assessment began with a simple question: was the expected health risk, what is the expected health risk from exposure to tricyclazole in rice at 0.2 ppm, and is this rice fit for consumption? To answer that, a standard risk assessment process was followed, starting by hazard characterization, and I won't repeat myself after what Dr. Eli has mentioned, and the second step is exposure assessment, and finally, risk characterization. This structured approach ensures that even when residues exceed the national MRL, regulators can determine whether the product actually poses a health risk to the public. We will start by reviewing the hazard value, then move to the exposure and risk characterization. EFSA have established toxicological reference values for tricyclazole. The acceptable daily intake, which is the ADI, is 0.05 milligram per kilogram body weight per day, representing the amount that can be consumed every day over a lifetime without appreciable risk. The acute reference dose is also 0.05 milligram per kilogram body weight, indicating the amount that can be safely ingested within a single day. These two numbers, both 0.05 milligram per kilogram body weight, are the scientific anchors for assessing any residue level of tricyclazole in food. In this assessment, those reference values provided the benchmark against which the estimated dietary exposure was compared. With those reference numbers set, let's estimate what an average consumer would actually be exposed to. As you can see on my screen, to quantify dietary exposure, three main data were applied from national consumption reports. And here, I want to emphasize on what my colleague also earlier mentioned about having reliable occurrence and consumption databases to boost the food risk assessment processes. The annual rice consumption equals 67.1 kilogram per person body per year, which translates to roughly 184 grams per day. The average adult body weight is 70 kilograms, and the raw to cooked conversion factor is 3, since residues are measured on raw grain, but consumptions data often refer to cooked rice. By calculating the daily intake of tricyclazole, which is the residue level, 0.2 ppm, multiplying by the daily consumption, which is 184 gram per day, the result is equal 0.368 milligram per day. Dividing this result with the average adult body weight, which is 70 kilogram, we get 0.0005 milligram per kilogram body weight per day. Expressed differently, the risk ratio represents 0.0105, which is nearly 1%. So let's translate those numbers into a clear conclusion for decision makers. At a residue level of roughly 0.2 ppm, the calculated dietary exposure represents only 1% of both the ADI and the acute reference dose. This means the exposure is approximately 95 times below the amount considered safe by international authorities. The conclusion, therefore, is ambiguous. The rice represents no appreciable public health risk under current consumption patterns. From a scientific standpoint, the product is safe for human consumption. The non-compliance arises solely from regulatory misalignment rather than toxicological concern. With the risk assessment showing no appreciable health risk, the next step was to define a proportionate, transparent risk management strategy. With the rice shipment was technically non-compliant, the scientific evidence indicated that the residue level posed negligible health risk to consumer. Given food security and supply chain consideration, 3 risk management options were proposed. First, to authorize a one-time release of the detained consignment based on the documented risk assessment results. This would prevent unnecessary food waste and disruption to the supply chain while keeping full traceability records. Second, to review the existing MRL for imported rice and consider alignment with internationally recognized standards such as New Zealand or Australia or any country that adopted the 3.0 ppm, a level supported by established good agriculture practices and risk assessments. Third, to develop a structured framework for evaluating and adopting import MRLs when justified by science. This framework would ensure that future decisions follows a predictable data-driven process. Together, these measures protect consumers, maintain public confidence, and minimize unnecessary trade disruption. Let's now look at what broader lessons this case study offers for our region. This example highlights an important distinction in regulatory science. First, regulatory non-compliance doesn't automatically equate to health risk. Second, this reinforces why scientific justification must guide MRL setting rather than relying purely on administrative or legacy limits. Third, a structured import tolerance or MRL mechanism helps bridge these gaps. It allows authorities to evaluate whether residue detected, although not aligned with domestic MRLs, remain within a safe exposure range based on a toxicological data and dietary intake models and international standards. Ultimately, adopting a data-driven decision-making framework strengthens both consumer protection and market continuity, ensuring food safety without unnecessary trade disruption. Let's always remember that science-based policymaking is the bridge between safety and trade, not a choice between them. To sustain such science-driven system, institutional capacity and cooperation are essential. No single country can develop all the required data alone. Our region shares common consumption habits, agriculture practices, climate conditions, and even export and import behaviors, making cooperation both logical and efficient. By sharing exposure and consumption data, residue monitoring results, and analytical databases, countries can create a joint foundation for harmonized import MRLs. Such collaboration would not only support trade among Arab states, but also enhances our collective position in global codex discussions. Ultimately, these efforts lead to stronger, more resilient food safety system. In conclusion, harmonizing import maximum residue limit is not only a scientific necessity, but also a policy opportunity. It allows our region to ensure consumer protection while promoting predictable, fair trade. The process begins with detection, moves through structured assessment, leads to proportionate decision, and ends with continual review. Import MRLs serves as a bridge connecting science and regulations until full harmonization with Codex is achieved. By anchoring our decisions in shared science, transparency, and collaboration, we can build food systems that are both safe and economically resilient. This is the vision to make the Arab region a model for evidence-based food regulation that protects its people and supports its producers. With that, I would like to thank you for your attention. Thank you so much.
In July 2025, experts from across the Arab region met in Muscat, the capital of the Sultanate of Oman, for the workshop on data collection and analysis for Codex proceedings. The event was organized under the Arab Codex Initiative by AID-SMO and G-Force, funded in part by the U.S. Codex Office and by Food and Beverage Regulatory Committee and the AmCham Dubai, and under the high patronage of the Ministry of Agriculture, Fisheries and Water Resources of Oman through its Food Safety and Quality Center. It was supported by CCNE, IU-FOST, GCC Standardization Organization, Université Laval, and the risk analysis platform Herrera. Over 2 days, 50 participants attended in person and 36 joined virtually. Building regional skills in dietary exposure assessment, GEMS food data submission, and setting maximum levels for contaminants. Practical exercises focused on lead in herbs and spices, mirroring real Codex procedures. Key recommendations included nominating national data focal points, standardizing data collection, sharing Codex priorities, and responding to JECFA calls for data. This workshop marked a major step in strengthening Arab engagement in global food safety.
You're very tall, and I'm sure. Thank you, thank you, Dr. Mustafa, for this important and insightful presentation. And now I kindly invite Professor Samuel Godefroy, President and COO of G-Force, and the President of the International Union of Food Science and Technology, IFOST. of course, to wrap up the outcomes of this session.
Thank you very much, Ruba. And again, thank you to all those who stayed with us at this late hour. We could not finish this session without reviewing some of the key messages that we've heard. I believe we got through this session a great illustration of the way food risk analysis, when applied, serves to support more robust food regulatory decision-making and supports more predictable and more trustworthy food control systems. The last presentation, I think, offered a great illustration about how a problem was turned into an opportunity. A problem in the form of an incident management situation triggered a reflection about the suitability of a regulatory limit, and applying a risk analysis process led to question this regulatory limit and to take into account definitely the public health considerations and consumers' health, which is of the utmost importance, but also food security considerations and ongoing flow of trade. So essentially moving us from an incident management situation to the point of making rules that are systematic and that are based on evidence, applying, of course, the risk analysis framework. The second presentation showed us how important it is to continue to collect data and to assess the risks associated with priority contaminants in food. Aflatoxins are major food hazards. In fact, they are amongst the limited number of food chemical hazards for which we have a clear association with the foodborne illness, in this case with the chronic situation that is hepatocarcinomas. So in this case, there is a need for continued monitoring, there is a need for continued application of mitigation strategies, particularly when we consider that this contaminant occurs in most consumed foods in a region such as the Near East region. And in fact, that presentation illustrated that to the question we don't have data, there may be another answer, because data seems to exist if you look for it. And even though, again, the study showed a number of limitations because it was based on published data, it nonetheless allowed to give an initial response and hopefully to trigger more interest in the collection of data. And this brings me to the beginning of this session, which really highlighted the foundational aspects that we need to pay attention to. Dr. Eli Bouyazbek, I think, gave a great overview of the areas where we need to pay attention and we need to continue to invest. And in fact, I would say that we are finishing with how we started this session. It is indispensable for any robust food control system, whether at the national level or at the regional level, that it relies on some key components that allows the decision making. We need to have data, we need to have competencies, and we need to have processes that allow to use this information in order to generate decisions that help protect consumers and that also enable to support the economies of the countries or of the region. Of course, resources are limited for any single country or any single jurisdiction to be able to fulfill those responsibilities alone. And that brings us to the importance of collaboration. A gathering such as this one is a great opportunity for us to reiterate the calls for collective action, the calls for coordination, and the calls for collaboration. We believe that there are some steps that have been taken in this region in particular. whether be it under the sub-regional organizations such as the GSO. We understand that the GCC Cooperation Council and under the auspices and the leadership of the Food Safety Coordination Framework that this union had set to itself is examining, in fact, opportunities for the creation of regional centers for risk assessment or regional structures that allow better collaboration and the ability to generate these scientific assessments that guide decision-making. We need to utilize these experiences and broaden them even further at the level of the region. So looking obviously at the Arab region, but also looking even broader where the Arab region does trade, whether be it with Asia or with some parts of Africa. These regions are definitely in need for continued investment and continued collaboration and coordination. Once again, I would like to thank all speakers for their contribution to these illustrations, and we hope that what we showcase today will serve in order to support further initiatives and further investments in food safety. Thank you very much.
The Global Food Regulatory Science Society. GFORCE mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control systems, Our interventions focus on promoting food regulatory scientific disciplines, consistent reliance on food regulatory sciences enables robust food control programs, contributing to more resilient food systems and a safe, accessible, abundant and nutritious food supply.